Welcome to the Principles of Pipetting. So what is a pipette? A pipette is an instrument designed to accurately transfer small amounts of liquid, anywhere from fractions of a microliter up to milliliters, and they can be used for a wide variety of applications, including PCR, DNA extractions, and water testing. The first pipettes invented were Pasteur pipettes. These were used by Louis Pasteur during his research in the 18th century. They were used widely and then eventually improved upon. Some of their limitations include that they are fragile, they have limited accuracy, they tend to be time-consuming, and can pipette only larger volumes of liquid. The next phase in pipette development are fixed volume pipettes. These are very precise, however, they have limited use. They are still in use today, and they can be used for very specific applications, specifically in GLP labs and any sort of student lab. Then the final evolution in pipettes are adjustable volume pipettes. These were invented by Dr. Warren Gilson in 1969. And the adjustable volume pipettes are very important, not only because they are accurate, but it also allows the technician to aspirate the exact volume that they want, not just what a fixed volume pipette would let them do. Pipettement pipettes are convenient, accurate, fast, and can pipette micro volumes of liquids all the way up to milliliters. So how does it work? There are two main types of pipettes, air displacement and positive displacement, and they function very differently. Air displacement pipettes have an air cushion inside the pipette that is displaced by the piston during aspiration. This means that the volume of the air displaced is equivalent to the volume of the liquid aspirated. Some examples of these standard pipettes are the Pipettement P, Pipettement Neo, and the Pipettement M. Positive displacement pipettes work like a syringe to aspirate and dispense samples. These are more specialized pipettes that are designed to handle problem samples. Some examples include the Microman and the Distroman. After you've picked out your pipette, you want to make sure that you organize your workstation. Adjust your chair or stool so it's the appropriate height. You should be able to rest your elbow comfortably on the surface and still pipette. Always work with your hands below shoulder height, and make sure that all necessary objects are within easy reach. So the most used objects should be closest to you, and the least used objects should be further away. Fitting a tip. There are two different methods for fitting a tip, one for air displacement pipettes and the other for positive displacement pipettes. For air displacement pipettes, hold the pipette in one hand, then gently press the tip holder into a tip. Next, use a slight twisting motion to seat the tip firmly on the tip holder. Make sure not to hammer a tip onto the tip holder. This can cause a number of problems. It can cause a bend or break of the tip holder. It can also cause a bend of the piston assembly, which will have an impact on your accuracy. And it can also be extremely difficult to eject a tip that's hammered on. For positive displacement pipettes, to fit a tip, you press the plunger button to the second stop to open the clamp assembly. Then fit the clamp onto the piston. While keeping the tip inside the box, depress the push button until you hear a click. If you're assembling your CP tips by hand, hold the tip in your hand after grabbing the piston assembly and push the button again until you feel a click. How to adjust the volume. You can adjust the volume with the push button and the thumb wheel for newer models of pipettes. For older versions, volume adjustment happens with the thumb wheel. So hold the body of the pipette in one hand, rotate the thumb wheel or push button with the other hand. To reduce parallax errors, either close one eye or turn the pipette horizontal. Mechanical backlash. By definition, this is dimensional dispersion from the counter and adjustment screw in the pipette. So what does this mean? It means that these parts fit together like gears. And because of this, variation in volume setting technique will affect the accuracy. So in order to improve reproducibility, always finish setting the volume clockwise. So to decrease the volume, turn the push button slowly to the desired volume, being careful not to pass it. Then to increase the volume, rotate the thumb wheel 1/3 of a turn above the desired setting, and then slowly turn back to decrease until you reach the desired volume. This way, mechanical backlash always occurs in the same position, improving your reproducibility of results. The pipetting sequence for an air displacement pipette involves 5 steps. Before the first step, make sure to pre-rinse the tip with the same liquid that is being dispensed. For preparation, hold the instrument in a nearly vertical position. Depress the plunger smoothly to the first stop position. Second step of aspiration, immerse the tip in the liquid, allowing the plunger to move up slowly to the rest position. Wait 1 second so that all the liquid has time to move up into the tip. Then for distribution, place the pipette at an angle anywhere between 10 and 45 degrees. Place it against the inside wall of the vessel and depress the plunger smoothly until the first stop. Then for the purge step, make sure you wait 1 second, then depress the plunger to the second stop position to remove any remaining sample from the tip. Then while removing the pipette tip, make sure to let it slide along the side wall of the vessel. And then for the final step, simply allow the plunger to move to the rest position, controlling the movement the entire time. The pipetting sequence for positive displacement pipettes involves 4 steps. For preparation, press the plunger button to the first stop, and the piston will move to the appropriate position. Next, for aspiration, immerse the CP in the liquid and release the plunger, letting it move up to the home position. Next, for distribution, press the plunger button to the first stop, and the piston will move down and expel the liquid out of the capillary. And lastly, for ejection, press the plunger button all the way down to the second stop. The capillary and piston are ejected without hand contact. Tip immersion depth is another thing to keep in mind because the immersion depth of your tip can have a significant effect on your results. If the tip is immersed too deeply in a sample, droplets will form on the outside of the tip and be deposited along with the sample. And if a tip is not immersed deeply enough, vortexing can occur and your pipette will not aspirate the selected volume. There are 5 steps to pipetting. First, make sure you organize your workstation. Then, make sure to fit a tip properly onto the pipette. Next, adjust the volume. Then, aspirate and dispense the sample. And finally, eject the used tip and store the pipette in an upright position. Storing a pipette in the upright position, make sure to protect it from possible.
Gilson or Finn micropipetters are one of the most commonly used instruments in a molecular biology laboratory. They're used in conjunction with plastic disposable pipette tips to measure or transfer small amounts of liquids, usually from 0.2 microlitres to 1,000 microlitres or 1 milliliter. Different pipettes work across different ranges of volumes and are named by the maximum volume they pipette. The different pipettes look very similar, so they're labelled on the top of each push button with a capital P followed by a number which indicates the maximum volume for that particular pipette. A P2 pipettes between 0.2 and 2 microlitres. A P20 pipettes between 2 and 20 microlitres. A P200 pipettes between 20 and 200 microlitres. And a P1000 pipettes between 100 and 1000 microlitres. A pipette is an expensive precision instrument. Improper use will damage the pipette, so correct use is essential. You should never use a pipette to measure volumes outside of its range. For example, using a P200 to pipette to lower volumes such as 10 microlitres or to higher volumes such as 250 microlitres will result in inaccurate volume measurements and will damage the internal mechanism of the pipette. Each pipette has a vertical row of 3 numbers visible in the body of the instrument. To set the volume, you can use either the thumb wheel or, on newer models, the push button. This will cause the 3 number dials in the body of the pipette to rotate and change the volume of liquid that will be taken up. However, the dial numbers indicate different volumes depending on which pipette is used. For example, these 3 pipettes have been set to read the same numbers on the dial, 152. For the P2, this means that the pipette is set to a volume of 1.52 microlitres. The bottom 2 dials are in red, indicating 5/10 and 2/100 of a microlitre. For the P20, this means that the pipette is set to a volume of 15.2 microlitres. The red bottom dial shows 2/10 of a microlitre. With the P200, the same visual setting means that the pipette is set to a volume of 152 microlitres. This P1000 is set to 0.52, which indicates a volume of 520 microlitres. A setting of 100 indicates the maximum volume for this pipette, which is 1,000 microlitres. The uppermost number is in red, which for this pipette indicates the number of millilitres. Once you've set the volume you need, attach the appropriate pipette tip to the end of the pipette. P20 and P200 pipettes use the same yellow tips. P1000 pipettes use larger blue tips. Another type of tip, called a filter tip, is often used in the laboratory. The sterile filter within each tip can help to prevent contamination. Hold the pipette with one hand, with the narrow side resting in the palm of the hand. Add the pipette tip by gently but firmly pushing the pipette into the pipette tip, which is held in a tip box. You may need a little practice to learn to apply the right amount of pressure to give a good airtight seal between the tip and the pipette. Liquid is drawn in and expelled using the pipette's push button. Gently apply pressure to the button with your thumb until you feel a natural stop. This is called the first stop. The distance you need to push the push button down will vary depending on the volume you require. A P20 set to 2 microlitres will require less push button movement than a P20 set to 20 microlitres. So to use the pipette, push the button down to the first stop. Then, keeping the push button at this level, place the pipette tip about 2 millimeters into the liquid you wish to draw up. Release the push button by slowly allowing it to return to its original position. Pause for a second to make sure all the required volume of liquid has been taken up into the tip. This is especially important when using more viscous liquids, which take longer to draw up. Withdraw the pipette tip from the liquid and place it inside the recipient container. Slowly push down on the push button. This will release the liquid in the pipette tip into the tube. This time push beyond the first stop. This ensures that any residual liquid is expelled from the pipette tip. Fully withdraw the pipette tip from the liquid before you release the push button. So to recap, push down the push button until you feel some resistance. Place the pipette into the liquid and slowly release the push button. Release the liquid from the pipette by pushing past the first stop to fully expel the liquid from the pipette tip into the recipient container. Withdraw the pipette from the container before you release the button. For very small volumes, the aspirated liquid can hang in a drop from the end of the tip and may not be transferred to the tube at all. So you should touch the tip to the inside wall of the recipient container whilst expelling the liquid. Once you've aspirated the liquid, release the pipette tip from the pipette by pressing down on the tip ejector. This is the small white button at the top of the main body of the pipette. Be sure that the attached pipette tip is inside the appropriate waste container before pressing the tip ejector. A new clean pipette tip should be used with each new liquid, or if the tip touches any surface or any liquid other than the one you're pipetting. If in doubt, change the tip. Do not use a pipette without a tip attached. Liquid should never enter the body of the pipette. It will cause the pipette to corrode and will be a major source of contamination between liquids and experiments. Do not use a pipette past its volume limits. This causes pipetting inaccuracies and also damages the pipette. If you're having trouble attaching a tip to your pipette, don't repeatedly jam the pipette into a tip. This can damage the pipette. And if the tip doesn't stay on the end of the pipette, simply repeat the procedure with a new tip. When pushing down the push button to take up a set volume of liquid, don't push past the first stop. If you push past the first stop, the volume you then take up will be too large. When taking up liquids, don't simply let go of the push button. The liquid can be sprayed around the inside walls of the tip and up into the pipette body, causing inaccurate volume dispensing and pipette contamination. Make sure you release the push button in a controlled manner. If you don't pause after you release the push button, there won't be enough time for the correct volume to be taken into the tip. Air will be taken in instead. Also, when you're withdrawing a large volume from a narrow container, make sure the tip stays below the surface. Whenever you have liquid in a pipette tip, don't lay the pipette down. Liquid can get into the body of the pipette, which causes cross-contamination, pipette damage and inaccurate pipetting. And finally, remember when pipetting small volumes, touch the pipette tip to the side of the tube to ensure the liquid is released into the recipient container. Gilson or fin micropipetters